Hello, everyone. It's time to get ready for another week of lab. So this week we're going to be looking at the uh, pigments in spinach. So this lab focuses on pretty much identifying the presence and identity of the colored pigments from a spinach extract. So we're going to be looking at five major pigments present in spinach. So two of them you're probably quite familiar with. These are chlorophyll. So chlorophyll A and chlorophyll B. So these are going to be your green pigments. But there's a couple other ones you might not be familiar with, such as phaeophytin. So phaeophytin is more or less chlorophyll, but with the magnesium removed, and at which point it becomes much more of a gray. Then we have the beta carotene, the orange from carrots, as well as xanthophylls, which are oxidized carotenes. So these are going to be the major ones that we're going to be looking for. You may not see all of these pigments in your system, but these are the big five we're looking for. And so an extract of these pigments has already been prepared by placing it in a nonpolar hexane solution before you ever enter lab. You're going to go ahead and try and analyze this extract. So we're going to do this by separating these components using chromatography. So you may be familiar with a basic form of chromatography, such as this coffee filter chromatography. So chromatography really works by separating components based on their intermolecular forces. And it does this by having two major components. First of all, a stationary phase, which is a fancy way of saying a surface, and then a mobile phase. So that mobile phase is going to tend to carry sample over and past the stationary phase. So as you can see here with the coffee cup calorimetry, that stationary phase is either just water or ethanol, or the mobile phase is ethanol or water, and the stationary phase is the cellulose filter. And really what's going to separate each of these components, so the colors in coffee cup calorimetry, or these two different organic components, is going to be based on the relative speed at which the sample moves. And this is going to mostly be dictated by the intermolecular forces. Would it rather hang out with that mobile phase, or would it rather hang out with that stationary? And that ratio will change the speed. If you like the mobile phase more, you'll move faster. If you like the surface a little more, you'll move a little bit slower. And so again, this thin layer chromatography, or TLC, what it does is it works by separating a set of samples over a polar stationary phase. So the stationary phase in this case is a silica dioxide surface, which again is going to be a highly polar inorganic surface, which we're going to run our materials over. So the sample is going to be placed on a line, going to be placed on a plate by go ahead and first marking where you place your sample with pencil and then spotting your sample on that line. Then you go ahead and place it in a chamber containing your developing solvent. So this is going to be a solvent which is going to act as our mobile phase and carry our sample up the TLC plate, up the TLC plate with, again, the species that prefer the stationary phase less, tending to move faster, and those that prefer it more are going to move a little bit slower. So big thing here is once you let that solvent reach the top of the plate, you're going to go ahead and remove the plate and mark where, however far that solvent front got. And what we're going to do is compare essentially the distance that these sample spots move compared to each other. And this relative distance each of these sample spots move is going to be dictated by the relative polarity of both the sample, but also the solvent. So let's go ahead and look at this a little bit more detail. So if I've got a sample that contains multiple components, we're going to actually see multiple spots. But we're also going to need to make sure that we choose a stationary, we're going to want to make sure that we choose a solvent or a solvent mixture, so essentially a mobile phase that has the furthest spread of these samples as possible. So what we're going to find is that if you have a given brand of TLC plates and a given developing solvent, each of these components is going to travel at a relatively given rate, being faster or slower. But one of the big problems is the solvent front may not move as far each and every time. Sometimes you may let your TLC plate sit longer, sometimes less. So what we really need to find is that relative distance that your solvent changes. And we do this by calling the ratio to front, which is more or less the distance the sample traveled versus the distance the solvent traveled. So what you would do is you'd go ahead and take a ruler, start from where spotting line is or where you place your sample, and measure how far the solvent traveled. And that would be the denominator. Then you'd measure to the center of the spot we care about. And that would be the distance that specific sample or substance traveled. And that ratio is more colloquially known as the RF factor. Now, if I've got since my stationary phase, or that TLC plate, is highly polar, as it's usually silica or alumina oxides, what we're going to tend to find is less polar substances are going to travel much further because they're not going to stick to that plate nearly as well as a more polar substance would be. And one of the things you may sometimes find is that if you use a non-polar substance, any polar solvents might not even move at all. Because why would I travel with that solvent when I can hang out with my plate right here? And so they'll stick on the spotting line. However, if you use a more polar solvent, all of your solutes or samples are going to travel further, non-polar or polar alike. And as a result, a major component of TLC is trying to pick the right solvent. So if our solvent is too nonpolar, again, we may find that our samples might not travel off the line at all. And so this leads to a poor resolution of all of my samples look identical. However, if the sample is too polar, all of these samples are going to want to hang out with the solvent more than the plate. And so they'll essentially stick with the solvent front and all move together. And at this point, we also have poor resolution. So just like Goldilocks, the correct solvent is going to be something that is not too polar, but not too nonpolar. And this will keep our samples in the middle. And when my sample's in the middle, we're going to tend to see much better resolution in between our various different points. Now, one of the problems that we have is that the appropriate choice of solvent depends on what solute. There is no universal solvent that will work for everything. So anytime you're examining a new system, you have to go ahead and double check to make sure that the solvent you're using is going to work. And this honestly needs to be tested experimentally. And again, the ideal form is going to keep all of those RF factors in between 0.4 and 0.7. However, do note that sometimes if you have both a very polar and a very nonpolar substance, you may have to go a little bit outside of those. But again, trying to keep all of your samples as much in this middle 0.4 to 0.7 zone is going to be the best option. And again, if you're conducting TLC, which again we can use to evaluate the presence of any number of solutes in a system, because it is fairly quick and easy, as you'll find in lab, the first most important thing you need to do is make sure you choose the right solvent. And so again, what we can do is make sure that we tailor the polarity of the solvent to make the best RF factor. So again, if you're looking for a nice nonpolar solvent, you'll usually start with alkanes or cyclohexanes, and then working your way more polar. On the high polar side, that's going to be water acetonitrile. So again, one of the things you may find is that a pure mixture of alkanes may be too slow, or a pure mixture of diethyl ether may be maybe too fast. So one of the things you can do is mix two different solvents together to go ahead and have a net solvent that's somewhere in the middle and maximizes your RF. And again, if you test the solvent and find you need to make your RF larger, go ahead and try and work your way down this polarity chart and go to something more polar. And again, if you need something less, need a lower RF, go ahead and choose something less polar or adjust your ratio accordingly. And so the goal of our experiment is going to be to determine what ratio of acetone and hexanes essentially are going to be the best for developing solvent for our plant pigments. And you might end up making and you'll end up making some use of this next week when we're actually going to be trying to separate the species, as this is a way to distinguish what what species you've actually separated. Now, when actually trying to prepare TLC plates, the first thing you're going to do is draw that pencil line somewhere around 1 centimeter from the bottom of the plate. And then you're going to place a dot in pencil where each of the samples go. We're going to typically do this with graphite, as graphite is essentially with a graphite lead pencil, because graphite is essentially too heavy to be pulled up the TLC plate. Then we're going to go ahead and use a TLC spotter to collect the sample. I'll be showing you how to make a TLC spotter in lab, but more or less it's a capillary tube where you place it in your sample, in the sample extract I give you, and it will suck up some sample, and then you'll go ahead and be able to spot it down on the polar plate, and it will suck the solvent right back down. And again, we are using, we've already suspended your extract in cyclohexanes, so thus the cyclohexane can rapidly evaporate off, leaving you with just your pigment. Then we're going to go ahead and place, then you're going to go ahead and prepare a sample chamber containing a small amount of solvent. In this case, you'll be preparing about 10 milliliters. You're going to place it in a beaker and you're going to cover with a watch glass, because a crucial piece of most of these solvents is that they do evaporate, and this will help pull the solvent up the plate. Unfortunately, this also means if you do not cover your plate, all of the solvent can potentially be removed from the system, and you have a lower concentration of solvent along the plate. So again, slowing the progression. Then you're going to go ahead and let that solvent progress until you reach near the top. So somewhere in between about a centimeter and a half a centimeter from the top. And then you're going to remove the plate from the chamber. And it's very important that we do so at this point, because if you let your solvent progress too far, it will carry your sample straight off the top of the system, and you'll no longer have an RF to measure. And again, then once you remove this plate, you're going to mark how far use the pencil to mark exactly how far the solvent front moved, and then mark the center points of each of your pigments. That way you can go ahead and measure each of your RF values. Now, one of the things you may find is a couple of these pigments may be a little bit hard to visualize. So one of the things you can do is use a UV lamp. So a UV lamp will essentially help you see the solvent line if you did not mark it correctly, but also may help visualize some of these harder to see pigments, as they will absorb light in the UV region. So these are going to be the crucial bits for this lab. So again, go ahead and make sure to prep your notebook and prep your pre-lab notebook and do the pre-lab quiz. And then again, when we're in lab, the first thing we're going to do is we're going to work in groups to identify the best developing solvent for each of these sets of plant pigments. So first thing everyone's going to do is make a TLC spotter. And as I said, I'll show you how to do this in lab. Then each set of partners is going to be assigned to make 2 TLC developing chambers. I'll tell you which ones once we get in lab, so we divide them evenly. So these are going to go anywhere from 100% hexane to 100% acetone and in between. And again, think about how you would prep these solutions, as we will only need 10 milliliters of them. So what piece of glassware will be the most effective? Then each group's going to run the TLC plates in each of these solvents. So not only the ones you've prepped, but also the other four. And then you're going to calculate the RFs for each of these solvent mixtures. Then you're going to sketch each of these plates, record your distances, and record the RFs in your notebook. So again, you'll have a sketch of the plates, all of the corresponding distances, and your RFs. As you will need these to conduct the worksheet summarizing this lab. Then you're going to go ahead and parafilm a vial of the extract and store it for next week. Next week, we'll be separating the extract into its crucial components. Once you're done with lab, don't forget to take post-lab notebook scans. And as a heads up, there will be a ChemDraw assignment already available on Canvas, and you're going to go ahead and submit this on Canvas by the end of next week. So never too early to get started. And this will essentially is a way that you can go ahead and draw good, convincing organic structures using a computer program. So hope to see you in lab, and best of luck preparing.